FDA have clear expectations about what analytical methods you have to use to demonstrate similarity?
I think in general they do, but their approach has generally been to leave it to the individual companies to define what specific methods are going to be useful for a specific product. I think anybody who's been in the biologics business for a while pretty much knows that there's a standard dozen or so or 20 methods that you're going to employ, and they're well known throughout the field. But I think, and I think the FDA is relying a lot on that. But I think for biosimilars, what the FDA has said is you're going to use pretty much the same methodology that the branded products have used, with the exception, as I mentioned earlier, that you're probably going to look at more specific and new analytical technologies that may not have been available when the original brand products, some of which are 10 or 20 years old, were developed. As a matter of fact, I was at a conference last year where somebody from the FDA specifically said, we don't really care whether you use the methods that the branded product used. We want you to use the best methods that are available today to analyze both your product and the brand material and do the comparison that way.
Oh, interesting. So it's kind of up to you then as the biosimilar producer to do perhaps a more complete characterization of the reference product than was done on that product when it first got its approval.
Sure, and one of the interesting things, and I've talked to some of my colleagues who are in other biosimilar companies, we get the feeling that by the time we're done developing a biosimilar, we may know more about the brand product than the brand product company knows, because most of the development work was done 10, 20 years ago, and they didn't have to answer the same questions that we have to answer. So I think that's an interesting part of being in this business, is that we end up knowing as much or more about the branded product as they do.
So that brings up another question. So even the reference product is going to have diversity from lot to lot. So when you're testing your product and the reference product, how do you know that you're getting samples of a wide range of lots of the reference product?
So that's one of the biggest challenges. There are ways to acquire the branded product in the marketplace, and what we try to do, and what others do as well, is to acquire lots of a variety of different ages. And a variety of different expiration dates, where they are in their expiration date. We try to test materials that are as fresh as we can get them and as late in their expiration as we can get them. We typically get products from both the US, the EU, and other parts of the world to compare them. But it takes probably a couple of years before you really know that you've gotten enough products and enough diversity so that you can define what the reference product profile really looks like.
And then given that diversity, you have to kind of come up with some profile, as you're mentioning, of what the reference product is, even though it is not 100% consistent, right?
Right, exactly. It's very different from the traditional generic, where you get one or two lots and you can characterize it, and they're pretty much all the same. They might have a slightly different impurity profile, but it is one molecule and you're looking at one molecule. Here, I like to, my analogy is, you have to figure out what the ballpark is before you decide what game you're going to play. So you have to look at the whole diversity of the reference product, and it does take a long time. And we've recently, we typically get a couple dozen or more materials over time that we analyze before we really think we know what the brand product looks like. And the other thing is, it can change over time because the process can drift. So sometimes you'll actually see differences. 1 year, 2 years later, you'll see subtle differences because the brand company may have made a slight change to their process and may have filed it with the FDA and shown comparability, but there might be a subtle difference in that.